talking about my plans for the next 15 years. I think it's not an exaggeration to say that most of the people coming to this conference are interested in phenotypic evolution in one way or another. And I'm particularly interested in three aspects of phenotypic evolution, and that's diversification, and integration, and innovation. I'm going to use two examples, one from animals, one from plants, to just illustrate those three aspects. Let's go. So diversification is really nothing to say. It's obvious, right? There's so many diverse, beautiful organisms. And birds, flowers, whatever you name. And then organisms don't just diversify randomly. They diversify towards a particular trajectory. Now, for example, the birds, when you look at the bird beaks, they are very diverse, and they're in more or less in adaptation to some sort of dietary source. And you could be an insect catcher, could be a honeysucker, it could be also a nutcracker. But to be able to do one of the things, you only evolve a beak shape or toughness like that isn't really enough. Because the beak is connected with the tongue, with the cranial structure, as well as the skull, right? So to evolve a beak like that, you have to also evolve your tongue and your cranial structure, as well as your skull, in the same direction. And it's the same thing for flowers. Now we see those beautiful flowers, and adaptation to different pollinators. But you can't just evolve the petals. The petal can evolve into a particular shape and particular color, and for a hummingbird or for a bee. But you also have to evolve the stamens and the pistils and all the reproductive organs in correspondence to the petal shape and petal color, etc. Otherwise, if you only evolve a beautiful petal color, but if your stamens and pistils are all messed up, that's not going to be a functional flower. So that's the problem of phenotypic integration. And then, of course, there's also the bird beak evolved as a larva structure at the base of the common ancestor of all the extant birds. And like flowers evolved as a larva structure in the common ancestor of all flowering plants. Now, how do those larva work, those larva structure actually evolve in the first place? Now, before I get to the innovation in the last bit, I want to just briefly mention what kind of questions I'm interested in about diversification and integration. Now, one of the things we're interested in is what are the genes? It's very simple. What are the genes responsible for the phenotypic variation? And what are their mode of action? It's not going to be a single gene controlling all the different beautiful forms, right? There got to be multiple genes. Most of the times they're polygenic. But what's their relationship? Are they additive? Are they synergistic? Or are they anticyclic to each other? And also, where are they on the chromosomes? Are they tightly linked in a so-called speciation island, so they can evolve all together once, or they are just randomly dispersed across the whole genome? And also, what's the role of pleiotropy, linkage, as well as selection? That what kind of role they play in making phenotypic integration possible? To address all these questions, I should warn you that I only have one slide or two about data. Everything else is my dreams. We use those beautiful organisms to address these questions. Now, just to be clear, I'm talking about the flowers, not other things on the side. And this is a group of closely related monkeyflower species. They are fabulous systems to study to address these questions. For one, it's obvious that they have a huge phenotypic diversification, right? Like they are genetically very similar to each other, but they're phenotypically very different. Now, the right flower species is a typical hummingbird species, and with its stamen, pistil, all extended out from the corolla. And then the bee species is a typical bee species. It has a lighter guide that tells the bee that here's my lighter, come out here, the yellow lighter guide, right? And it has a landing platform for the bee to hook onto. And then there's also a sulfur. Now, everything just evolved into a very reduced fashion. The color becomes inconspicuous, the stamen, pistil become very close to each other, and the whole flower becomes really small. And the lighter is all gone, because they don't need pollinators. The reason that this system is really great, because although they are phenotypically very different, but you can cross them very easily. You can do a lot of interesting genetics with them, and you can cross all the four species, in fact, with each other, generating beautiful F1s. And you can also have those beautiful F2 population, segregating all those phenotypes, all the individual traits, are segregating in those F2 populations. Now, on one hand, this is great because those traits are segregating, so we can potentially do genetic mining. On the other hand, this is also caused a huge problem because you can tell most of those floral traits are quantitative in nature, which means they're all controlled by multi-genes. Now, how do we figure out what are the genes responsible for those? How do we even isolate each of those single locus and then look at what's that relationship, etc., and then where are they on the chromosome, etc., right? So this causes actually a huge problem. Now, the way I think it's the only way to solve this is to reduce the complexity by making so-called dual isogenic lines. Now, the idea is that you start from two different species, and you make a cross, and you keep back crossing to one of the species. Okay? And then by back crossing, you can basically gradually clean up the genomic background of this so-called dual isogenic line to make that it's the same as one species, but only have one little chromosome chunk coming from another species. But of course, by making those dual isogenic lines, you are selecting them based on phenotypes. Okay? You're selecting those interesting phenotypes. You won't have the individual looks just like one species, except one trait is different. One trait that looks like more like the other species. So we sort of have started already doing this years ago, and those are two color genes. So that's the bee species and the hummingbird species, and we'll have this one locus for the raw intensity that controls the anthocyanin concentration, to the darkness of the pink. So we will integrate only just one gene from the bird species to the bee species. You can say you make the flower color much darker, but otherwise the whole plant looks just like the bee species. Similarly, the other dual isogenic we have is the gene controls the presence versus the absence of that white-green in the chlorophyll. Okay, so the bird species don't have that, and then when you replace that locus, you generate a flower that is just like the bee species, except now you don't have that green anymore. But you might ask, okay, so if we already started doing this years ago, then why this is still a dream? Now, to address all those questions I just mentioned, it needs a systematic effort. You can't just, those two flower colors is two of the many traits we're talking about. Okay? And even those two loci, those two genes, and those two dual isogenic lines took us 10 years to do. So that's why we couldn't really do those things like systematically, to dissect those like 50 or 100 different traits in this way. Until a few months ago, we finally have actually beautiful chromosome level genome assembly for all these guys. And now we can actually have, you can have endless amount of markers you want, so you can do genetic mapping much more easily. And once you make those dual isogenic lines, you can just sequence the dual isogenic line, and you can compare with the printed genome, and you can tell exactly which chromosome fragment is coming from, right? And also you can then start answering the question that where the genes are actually controlling those floral traits actually located in the chromosomes. Are they located in those random positions on the genome, or they may actually locate in those reciprocal translocated fragments. Now, if there are two genes controlling two different floral traits, if they're located in an inversion or in a translocated region, they are not going to recombine, and they're going to evolve together. So that has something to do with phenotypic integration. And those are kind of questions or conclusions we couldn't really draw before without those chromosome level genome assemblies. So to just demonstrate the utility of those genomes, and I started doing this, started a systematic effort about 6 months ago. And those are the sulfur, and this is a hummingbird species, and the F1 looks just like intermediate. And after just one generation of backcrossing with like marker assisted selection and phenotype, I already got one dual isogenic line that looks just like the sulfur, except the only difference is the flowers are a little darker. And also, just to show that flower color is not the only thing we work on, this other dual isogenic line looks just like the printer species of the sulfur, but the flower size is bigger. But you can tell that this is only about a 2-fold difference, but when you compare the bird species and the sulfur, they are probably like a 5-7-fold difference in terms of size, right? So we know there are other genes. We're just isolating one locus in this case. And you can also do that in the other background, of course. You can background that to the bird species, and we'll have this dual isogenic line. When you look at the front, they look almost just like exactly the same. But when you turn it back, you can tell that the corolla 2 the stamens and the pistils, they're all a little bit shorter. Although the petal lobe is actually exactly the same length. So you can already conclude that the petal lobe and the corolla and the corolla tube and the pistil and stamen length, those things are developmentally regulated, are decoupled. They are not regulated by the same gene, right? However, the corolla tube length and the stamen and pistil length, they could be controlled by the same gene. Or it could be controlled by multiple genes just sitting in a very tight linkage group. So how do we tell them apart? Now the problem is that if they are sitting in a chunk of chromosomes that don't really recombine, we also can't find the gene by genetic marking. So that would be an issue, right? And in most systems, in this case, you wouldn't be able to tell those two possibilities apart. Now that comes back to the dark side of my life. We also have a lot of chemically induced mutants and transgenic tools for this whole system. So when I say dark side of my life is because it's much harder work. And you can actually figure out a lot of the basic genetic and developmental control of those individual floral traits by doing hardcore developmental genetics work. And then you can use that as your candidate gene toolbox to actually test if you map the three traits into this one interval, and now let's say 500 genes in it. But you can tell, okay, based on our developmental genetic work, there are 2 of those genes are likely to play a role in this, and you can do transgenic manipulation. So this is another species, we can also do that, and this is just to show you we can do a lot of transgenic work in this. So I think actually with all the resources and tools, I think in 15 years maybe we can actually answer questions like what are the genes responsible for the phenotypic variation, and what are the mode of actions, additive, synergistic, or epicyclic, and where are they in the genome, and how do they actually contribute to the phenotypic diversification and integration, and what's the role of pleiotropy, et cetera, et cetera. And I'm going to skip the innovation part. Oops. I'm just going to, there we go. I just want to actually acknowledge that my lab group there, they are really the ones who kept the life interesting, and I want to acknowledge the funding resources actually kept my dreams alive. Thank you.